Glycolysis. Glycolysis, glycose meaning sugar and lysis meaning splitting. We all know that. So glycolysis is the splitting of sugar and it occurs in the cytoplasm of both aerobic as well as the anaerobic animals. And when we say anaerobic animals, we don't mean to say viruses, because viruses, they do not have cytoplasm. So glycolysis, it was discovered by Gustav Emden, Otto Meyerhof and J. Parnas. Therefore, it is also called as the EMP pathway. So when summarizing the glycolysis, it is very simple. What is it? It is the partial oxidation of glucose into two molecules of pyruvic acid. So let's start the process. So as we all know that photosynthesis, we know all photosynthesis. What is photosynthesis? Okay, so photosynthesis, the end product of photosynthesis, we get the sucrose. Okay, we get the sucrose, and sucrose, it is converted into glucose and fructose with the help of enzymes invertase. Okay, and invertase, it is also called as sucrase. Now let's start the main glycolysis process. So first we have glucose. Glucose, it gets converted into glucose 6 phosphate. Now glucose was there, but 6 phosphate group, it is added to the 6th carbon of glucose. Glucose has how many carbons? Glucose has 6 carbons. Okay, so on the 6th carbon, phosphate group is added. Now where did this phosphate group come from? It came from ATP. ATP was triphosphate. Now after donating its phosphate group to glucose, it becomes ADP. Okay, so here we see that there is the utilization of ATP. Now fructose 6 phosphate is made from glucose 6 phosphate by the help of enzyme isomerase. This is very easy because these two are the isomers of each other. Therefore, again, this fructose 6 phosphate, it utilizes one more ATP. Now what is the function of ATP? It is to give phosphate group to this compound, that is the fructose 6 phosphate. So after receiving ATP, fructose 6 phosphate, it gets converted into fructose 1,6 bis phosphate. Do you realize how this got converted into fructose 1,6 bisphosphate? It obtained one phosphate group from ATP. Therefore, it gets converted into this one. Now, fructose 1,6 bisphosphate, it gets splitted up. Okay, it gets splitted up. Now you can see that fructose 1,6 bisphosphate, it gets splitted up into 3-phosphoglyceraldehyde and dihydroxyacetone phosphate. Here the enzyme uses aldolase, but it is not necessary to remember the enzymes names. Okay, so here 3-phosphoglyceraldehyde and dihydroxyacetone phosphate are again isomers. Okay, so with the help of enzyme isomerase, they can get interconverted into one another. Okay, so till here from glucose to fructose 1,6 bisphosphate, it was a 6 carbon compound, but when it got splitted up, okay, when it got splitted up into 3-phosphoglyceraldehyde and dihydroxyacetone phosphate, now they became 3 carbon compound. Now these two are 3 carbon compound. Okay, now since these two compounds are interconvertible, dihydroxyacetone phosphate can get converted into PgAl, and these processes which we are going to study, it can occur to this compound also. So we'll discuss about this again in few minutes. So here we have fructose 1,6 bisphosphate making this 2, and this product, that is the 3-phosphoglyceraldehyde, that is also called as PgAl, this gets converted into 1,3-bisphosphoglycerate or 1,3-bisphosphoglyceric acid. Okay, so here we saw that phosphoglycerate. Okay, phosphoglycerate. Okay, so bisphosphoglycerate, there is addition of one more phosphate group. So uh, for addition of any phosphate group, we saw that there was the need of energy. So here the energy it is given by NAD plus. Okay, and by receiving hydrogen from this compound, that is PGAL, it gives hydrogen, it gets added to this one. So NAD plus, it gets converted into NADH plus H plus. Okay, and this NADH plus H plus, or NADH2, it later on goes to the ETS cycle, that is the electron transport system. Okay, and there it makes three molecules of ATP. Now 3-phosphoglyceraldehyde finally made 1,3 bisphosphoglycerate, which is also called as BPGA. Now this bisphosphoglyceric acid, it has to make 3-phosphoglycerate. Now you see what is happening. Bisphospho, it is from two phosphate groups, which was here, it is converting into one phospho group only. So here what happened to the one more phosphate group which was here? So the phosphate group which was here, it goes to ADP. Now ADP receives its phosphate group, gets converted into ATP. So here this is the first synthesis of ATP in the glycolysis, which is from BPGA to 3-phosphoglycerate. Now 3-phosphoglycerate gets converted into 2-phosphoglycerate by the enzyme mutase. So we can see that this is just the shifting of position of the functional group from 3 to 2. So mutation. Okay, so mutase is the enzyme which helps in this conversion. So 2-phosphoglycerate, again it has to get converted into phosphoenol pyruvate. Phosphoenol pyruvate. Now when we see pyruvate, we are very closer to the last, that is the end product. Okay, so for 2-phosphoglycerate, with the help of the enzyme enolase, it gets converted into phosphoenol pyruvate. Very easy. 2-phosphoglycerate gets converted into phosphoenol pyruvate with the help of the enzyme enolase. Now when this enolase enzyme acts upon 2-phosphoglycerate, one water molecule is removed and phosphoenol pyruvate is formed. Now phosphoenol pyruvate, again it has to lose this phosphate group. So where does this phosphate group go to? It goes to the ADP, which is the diphosphate. It receives one more phosphate group and therefore makes ATP. Okay, so here the enzyme is the pyruvate kinase, and it makes the pyruvic acid, which is again 3-carbon compound, because from here till here this is the 3-carbon compound. Now as we talked about it earlier, dihydroxyacetone phosphate also gets converted into 3-PGAL, that is phosphoglyceraldehyde. Therefore this also makes 3C, that is the 3-carbon pyruvic acid. Okay, pyruvic acid is 3-carbon, it also makes pyruvic acid. So this same process, if it gets converted into this one, isomerase is used. Therefore after using isomerase, it gets converted into PGAL. Therefore it also makes pyruvic acid. So this is also making pyruvic this is also making pyruvic acid. So one pyruvic acid from DHAP and one pyruvic acid from PGAL, it makes two pyruvic acids. This pyruvic acid. Now what will happen to this pyruvic acid? As we talked about earlier, about it. Okay, so this pyruvic acid, as we said, that glycolysis, it is only the partial oxidation. Okay, it is only the partial oxidation, which means that all of the energy that is required for carrying out the normal processes in the body, it is not enough by only the glycolysis process. Therefore, this pyruvic acid, it goes to alcoholic fermentation, okay, goes to anaerobic respiration and to the aerobic respiration according to what animal it is. So, pyruvic acid goes to those processes. So, the ATP used in glycolysis process is first here and here. So, 2. ATP used is 2 and ATP made is 4. Why? Because here 1, here 2, and from here also the same process is going to occur. Therefore, 1 plus 2, 1 plus 1, 2, and again here 1 plus 1, 2. So total is 4. So net gain of ATP is 2, but NADS2 that is being produced over here, it also goes to the ETS, that is the electron transport system or electron transport chain later on, and makes 3 ATP molecules. So this is all about glycolysis.